We are with our first question, where you can see some statements regarding the 30 nanometer DNA chromatin fiber structure. Here I am stating the most common structure of the DNA, that is the most uncondensed, means the relaxed from the DNA whose diameter is 2 nanometer. Upon addition of the histone proteins, it converts into the bits on string structure, where these are the histone codes except the H1, and it wraps around the DNA. For its specific kind of look, it is known as the bits on string structure. Now, upon addition of the H1 protein, it converts into the 30 nanometer structure, where two kind of structure is possible. The one is the zigzag pattern, where these are the histone codes, and this one is the H1 protein. So, upon addition of H1, it converts into a zigzag model in the zigzag form. This is the 30 nanometer structure upon addition of the H1 protein with the bits on string structure. And the next one is the solenoidal model, where those are the histone codes. This is the H1 protein, and those are the linker DNA, which are linked like this. And the DNA is also stays wrapped around like this. So now come to the statement, where they are saying in the solenoidal model, the linker DNA connects the consecutive core particle. Yeah, in the case of solenoidal model, the consecutive core particles are connected via this linker DNA. So this one is the right answer. And for the zigzag model, the alternative codes are connecting together. So in the case of the second statement, this one is also right. And for the fourth statement, they are saying the H1 histone is essentially required as for zigzag model, but not for the solenoidal model, which is not true. If you want to make the 30 nanometer form, where these zigzag and solenoidal, those both forms are 30 nanometer form, 30 nanometer structure. So to make this structure, the H1 histone protein is also required. That is very much essential. So this statement is also wrong. And the last statement is stating about the H4 histone. This histone is present in this core, and that is very much essential for epigenetic regulations. So for higher order or the lower order folding, in both the cases, this H4 histone is very much required. So this statement is also wrong. So here the third option is the right answer. So here are some statements regarding the fidelity of DNA replication. At first, we have to understand what is the fidelity of DNA polymerase. This is the property of DNA polymerase, and this defines how accurately it is incorporating the nucleotide when it is doing its activity during the DNA replication. Let's see here the polymerase is present, and if it is incorporating the correct nucleotide, means the exact complementary sequence of this template strand, that means this DNA polymerase has the high fidelity rate. And if it is incorporating the wrong nucleotides against this template, that means its fidelity is too low. And let's see the first statement. It is stating about the 5' to 3' exonuclease activity. This exonuclease activity is responsible to remove the RNA primer. So it removes from the 5' to the 3' direction, and it is the property of DNA polymerase 1 that is not truly related with the proofreading activity of DNA polymerase. Instead, the 3' to 5' exonuclease activity is responsible for the proofreading property of DNA polymerase. So, this 3' to 5' exonuclease activity is responsible for the fidelity of DNA replication, but not the 5' to 3' exonuclease activity. So, this one is wrong. And the next statement is saying about the imbalanced intercellular concentration of 4 DNTPs. If the concentration of DNTPs are, if some DNTPs are in too high concentration and some are into low concentration, then it can enforce the DNA polymerase to incorporate the wrong nucleotide instead of the right one. So, so the imbalanced intracellular concentration could be a factor to decrease the fertility of DNA replication. And this imbalanced nucleotide concentration is intentionally used in the case of error-prone PCR. So this statement is totally true. The next one is the increased intracellular concentration of RNTPs. Also, increased concentration of RNTPs can be a factor to enforce the DNA polymerase to incorporate RNTPs instead of DNTPs. So, this although the ribonucleotide excision repair pathway is possible, but it has no relation with the proofreading activity of DNA polymerase. So, this statement is totally true. And the last one is stating about the removal of incorporation of the nucleotide. Yes, if there is any mismatch present in the DNA, if it happens via the activity of the DNA polymerase during the replication, it can be removed by the mismatch repair system. So that is very much useful to increase the fertility of DNA replication. So this statement is also right. So the option 2 is correct. And see some certain statements regarding the telomerase enzyme. So telomerase is generally known as the end part of the chromosome. Suppose this one is the 5 prime to 3 prime. Suppose this one is the mother strand and this one is the newly synthesized daughter strand. In the case of daughter strand, you will be able to see a RNA primer at this position. So after the successful replication, this primer should be removed because this is made via the RNA. So now there you can see a 3 prime overhang. So this overhang portion is made by single stranded DNA, which is very much prone to be degraded via the DNase enzymes. So if that happened, then we will start losing the information that was present at that site. So which is not that much favorable thing. So now here the telomerase enzyme comes to play. It is a special kind of enzyme that is having a RNA primer sequence that is inbuilt inside it, which has a uh, predetermined sequence, predetermined sequence with which varies depending on the species. And this enzyme also has a activity that is known as the RNA dependent DNA polymerase. So this will come here and gives it primer here, gives the primer here, and now you can see like this will be the 5 prime and this will be the 3 prime. So it can extend this way to fill that portion, and this portion will also be going to fill. And now you will see this primer is also made up of RNA. So after the successful replication, after that successful DNA RNA dependent DNA replication, so this part will also be removed. Now the next batch of telomerase enzyme will again come, and this will again extend that part, and the extension will go till a certain point. Still, even after the extension, you will not be able to restrict it from forming a 3 prime overhang. At least the 3 prime overhang will be there anyway. But in that way, it will be successful to restrict our genetic informations. So here, some proteins that is the telomerase binding proteins, those are known as like the cell-terrain protein, they can come and bind in that end regions to restrict the DNA from the degradation. So here, this kind of telomerase enzyme is generally present in the case of stem cells. But if they are present in the differentiated cells, what would be the problems? In the case of differentiated cells, they have a predetermined lifespan. If this kind of things are happening there, so they will not be able to go to the apoptosis. That will subsequently increase the number of cell, and the undifferentiated cells and that differentiated cells number will be too much high, because the apoptosis will be blocked. So that will be the unconditional growth of the cell, which may lead to form a cancerous situation. But if in the stem cells, in the case of stem cells, they are too much needed because they are having a very long lifespan. But in the case of stem cells, if the amount is getting too much low for them, then the disorder like the progeria can happen. This kind of disorder is related with the aging before the actual age. So now let's see the options. So here I also told about the TBP are believed to shield telomeres from the cell's DNA repair machinery. That is totally true. Next one is the telomeres in human cells are repeats of TTA GGGG. That is not true. This is the repeat of TTA GGG. 1G is very higher. 1G is not in the list. And this sequence that can extend up to 150 kb. 150 kb is too long. That is not energetically and evolutionary favorable. So in general cases for the human cells, it spans between the 10 kb to 4 kb. Actually, that 10 kb is very rare case. It happens for the very rare case. But in general case, 3 to 4 kbs you can see. So this is not right. The next one is in differentiated cells, telomerase is inactive. That is true, and leading to shortening of the telomeres. Yeah. So the shortening of the telomeres will happen over the hundred of cell divisions, and the damage which will perform the damage to the ends, and eventually it will go to the apoptosis. Like I told you here, no? So if that thing is happening again and again, there is no telomerase enzyme, and they are loosening this part depending on each cell division cycle. So after 100 cycles, they will lose a huge amount of DNA and huge amount of information. So this one comes true. The last one is the persistence of telomerase activity in several cancers allow the cells to continue to proliferate. That is true. So the option is A, C, and D. So fourth option is correct. So this question is about two operons. One is lac operon and another one is trip operon. Whereas the lac operon is the catabolic in nature. That means when the ligand is present, then it will produce the outcomes, the proteins. It will present in the mRNA. But in the case of trip operon, it is the anabolic type of operating system. So in that case, if the ligand is present inside the cell, then it will reduce the expression. So here, so this is the structure of, I am drawing the structure of lac operon. Here you can see the repressor protein. This is the mRNA, and here you can see promoter, operator, and there the lag genes means Z, Y, and A. So here the repressor protein is producing from this part, and it is going and bounding to that way, and it is not allowing the RNA polymerase to go on. So here the transcription has been blocked. But if lactose is available inside the cell, it is producing that lactose is producing into allolactose. That is the ligand for this lac-I, that lac repressor, and then allolactose can go and bind with it, and so that it will be removed from here. So in the case of lac operon, in the normal condition, when there is a deprive of that allolactose, then the production of these enzymes are too low. But in that ligand condition, the production is too high, because that time the repressor is not present there. And what is the structure of the trip operon? In the trip operon case, suppose this is the trip repressor, trip R, and here you can see the trip promoter, trip operator, and the rest of the genes you can see, that is trip E, D, C, B, and A. And here from the trip repressor, a protein is getting synthesized that is known as the aporepressor. Aporepressor, which stays in monomer form and thereafter converts into the dimer form. In general cases, it can't go to the operator, and so the this system stays on when the trip is not present there. The tryptophan is not present there. So in the case of trip R, in normal condition, it stays on. So the production is high. But if that ligand means the tryptophan comes and bind to this dimer, this uh, operator, this aporepressor dimer, it comes and binds with the dimer, the tryptophan, then there a certain conformational change happens, so it goes and binds with this operator, and now the transcription is no longer possible. It is not possible. So in the case where the ligand is present, the repressor is not there, it is too low. And now come to the question part. So here on the A, it is saying that allolactose bind to lac I leads to its poor binding to the lac operator. Yeah, that is totally right. So in general case, the lac I would be there at the operator site. But if it binds with the allolactose, then it will be removed like operator. Whereas the tryptophan binding to trip R leads to its better binding to the trip operator. That is also true. So the option 1 is right. In the next option, the allolactose binding to lac I leads to induction of lac operon. That is also true. Yeah. So when that lac I is coming and binding to the allolactose, then the induction of lac operon is happening. Yep. And whereas tryptophan binding to trip R leads to repression of the trip operon. So from this figure you can find it out. So that is also right. The next option is the binding of allolactose and tryptophan to lac I and trip R respectively leads to depression of their corresponding operons. No, that is not making the depression for both. For one it is depression, and for another one it is the activation. So that is wrong. And here also the binding of allolactose and tryptophan to lac I and trip R respectively leads to activation. So not activation, either it would be the depression or the activation. So the option 1 is correct. Here in this figure, you can see 3 plates where the plaque assay was performed, and this different result is related to the different type of bacterial strain and viral strain. And here in the first plate, you can see the number of plaques are too high. So here the infection is high, here the infection is relatively moderate, and here the infection is not present at all. So this kind of combination of bacteria and virus is responsible for this kind of result. So here you can see two kind of bacterial strain. One is the B strain and another one is the K12 strain. So both are the strain of E. coli bacteria. The B strain is more potent to the infection cause its cell membrane permeability is relatively high than the K12, and it lacks in the presence of some OTS, whereas the K12 is relatively more resistant to the infection. And that is all from the bacterial part. And the next one is the viral part. It is stating about the T4 fudge. and it is saying about the R2 locus. This locus in the first genetics, this locus is very much important to determine whether this bacteria is going to do the lytic or lysogenic cycle. So, and it also determines the host range of the bacteria of this virus, this T4 virus. So here the wild type T4 first, its host range is relatively higher than that of the mutant one. So it can infect and form the and can generate and can perform the lytic cycle in the case of B and K12, both bacterial strain. But the mutant form only can uh, form the, it can infect both, but it only can form the it can only perform the lytic cycle in the case of B strain, because it is more potent in the more potent to the infection. So here in the first figure, the amount of infection is too high. That means it must be the B strain, and the third one is with the less infection, so it must be the K12 strain. And here the middle one must be the both, the either the B or K12. It can be B or K12. And here the now let's go for the viral part. Yeah, for the high infection, so in the first figure, so it can be wild type and mutant both. And for the next one, because here only because to infect the K2 strain, we need the wild type. So in the second case, it must be the wild type of virus. And the last case, here no visible infection is present, so it must be the mutant one. So depending on the combination, it says the second option is correct. So here this question is on the E. coli's SOS response to DNA damage. So first of all, the conventional DNA repair mechanisms work before the DNA replication happens. If that fails, or the exposure to UV rays or mutagens during the DNA replication, that can induce the pyrimidine dimer or certain kind of lesions. Like if this is the DNA and DNA, this is the replication fork. If the DNA polymerase is going through, if they are getting any kind of mutations, the pyrimidine dimer or certain kind of lessons, if they get it here, then they will not perform. They can detect it and they will stop the replication. To proceed the replication, we need certain kind of enzymes. So this kind of enzymes are known as the translation polymerases. So one of the example is the DNA polymerase 5. This kind of polymerase will come here and replace this DNA polymerase, and they will continue the replication, and thereafter, after passing that place which has some maybe some pyrimidine dimer or certain kind of lessons, after passing that way, it will itself replace it, and thereafter again they will recruit the DNA polymerase to go on the polymerization. And in this kind of lessons or this kind of dimer formations, we need the translational polymerase to overcome this stalled condition of DNA polymerase, so that at least the DNA replication can happen. So in the SOS response system, the activation of the synthesis of translation polymerase is very much important. And even in the case of double strand break, if there is a double strand break, here you need the REC-A protein to continue the repair via the SS pathway. Here, this works on the homologous recombination pathway, which is the most convenient to repair the double strand break. But here is a catch: the REC-A enzyme can't bind on the double stranded sequence. It needs a 3' overhang to bind over there. So if that is like that, then only they can come and bind. So to do that, we need some exonuclease which can chew away the 5 prime of the 5 prime end of the DNA and can form a 3 prime overhang. So this rake enzyme will bind here and from the homologous recombination. But to do so, but to do this recombination, so do this recombination, it will bring another DNA strand which is conformationally homologous to this sequence, and this will recombine. So here to do so, we need some SOS response regulatory genes which can only be synthesized if there is no blockage in the promoter. So here the Lex A works as a repressor. So what is the work of the that Lex A repressor? So in the case of SOS response regulatory gene, if those are the SOS regulatory genes, so at the operator site, the Lex A repressor binds and it blocks the translation, the production of this kind of proteins. So no protein is produced uh, in the normal case. But if there is an increased concentration of Reke protein, then it will produce the auto catalysis. It will produce the auto proteolytic activity. It is related with the autoproteolytic activity of this Lex A repressor protein. The Lex A repressor will break here down and it will removed from the repressor so that the translational machinery can access these SOS response regulatory genes, which will further effects here, and so the DNA repair can happen. So this is also right. And so breaks. So RECA is activated due to the blunt ends of double strand breaks caused by DNA damage inducing agents. That is not true, because I have told that we need a 3 prime overhang. And the last one is also not true. The destruction of Lex A promoter synthesis of photolyase. There is no actual relation with photolyase with the Lex A repressor, but this photolyase is a very responsive enzyme in the case of pyrimidine dimer formation process. When we want to remove that pyrimidine dimer formation, then the photolyase enzyme is very much essential. But here there is no relation. This photolyase is not a part of SOS response regulatory gene. So the correct answer is option 3. This question is stating about two bacterial fudge. One is lambda and another one is the P1 fudge. So here it is mentioned that both the fudges are temperate fudge. What does that mean? Temperate fudge means who can successfully infect in both the way. One is the lytic way and one is the lysogenic way. In the lytic way, after the successful transduction, this is the bacterium cell, this is the bacterial genetic material, and this is the fudge virus. And it is incorporating its genetic material inside the cell via the transduction. And here are the here are the viral genetic material. And if the condition is in favor of the virus, then it will transform into the lytic cycle, where it hijacks the bacterial cellular machinery for high amount of translation, replication, these kind of things. And within no time, it produces its functional proteins and other structural proteins and replicates its genetic material. And thereafter, it lies or break down the cell's membrane, the bacterial membrane, and goes outside for the next cycle of the infection. And they all are having their own genetic material. But whether if the condition is not in favor of them, then they will transform into the lysogenic cycle. What is the lysogenic cycle? In that lysogenic cycle, the viral genetic material, it can be incorporated inside the bacterial genetic material or the chromosome, or else it can stay outside the bacterial chromosome as a as a low copy number of plasmid. In the case of P1 fudge, it stays outside the bacterial chromosome as a low copy number plasmid. But in the case of lambda fudge, here in that bacterial chromosomal DNA, there is a site that is called ATT lambda. This site is the attachment site for the lambda fudge, because this attachment happened in a kind of homology dependent attachment. This this lambda fudge's genetic material is also has a specific kind of sequence that attaches with this sequence via the homology dependent recombination. And now go to the option parts. So the first one is the here you have to identify the incorrect one. So both the lambda and P1 fudges are double stranded DNA virus. This is correct. And the next one is in their lysogenic state in E. coli, while the lambda fudge integrates into the genome, yeah, via the ATT lambda site, it integrates into the genome. In the case of lysogenic cycle into the genome, the P1 fudge remains as low copy number plasmid. It stays outside because it is not having any kind of attachment site or any kind of sequence homology. So this is also right. The next one is in their lysogenic state, both the lambda and P1 fudge are integrated into the genome in E. coli. That is wrong. And the last one is saying that in the lytic cycle, both the fudges occur in plasmid form in the E. coli. Yeah, in the case of lytic cycle, it never integrates into the bacterial chromosome because they will not get that much of time. This one is an experimental type question. So let's see what is the micrococcal nucleus. So MNase is an enzyme. It is a kind of endoexonuclease which works in the presence of Cr2 plus ion. That means Cr2 plus is the cofactor for this enzyme. Let's see if you have used the MNase on a chromatin like this. So, cause this one is a endoexonuclease, so it will come and cut this kind of area which are accessible for them. But they are not able to cut at this kind of sites because they are surrounded via the histone protein. But which portions are accessible, they can bind to that portion and cut that places. But in that case, this is a free accessible place for NMS, so they can for MNS come and cut this site. But here, if some other kind of proteins like DNA binding proteins or certain kind of other things are coming and binding to these accessible portions, that will no longer be the accessible for the MNase. So that in that case, it will not be able to cut the site. And depending on the question, the researcher has prepared 4 test tubes where that person has introduced this chromatin. Suppose this chromatin he has introduced and 4 tubes. And he also has used that MNase enzyme and he has treated the one is for 1 hour, other one is 3 hours, 6 hours, and 12 hours. Thereafter, he has run the southern blotting, this one and this one. The first one is correspondence to the probe 1 and the next one is on the probe 2, where the probe 1 and probe 2 correspondence to different location of the chromosome. And there, this is the result. In the probe 1 case, even depending on the time, the change of the concentration of DNA is too low, where you can see the rapid decrease in the concentration in the case of probe 2. What does that mean? Might be in the case of probe 1, this portion is of that part. So, which part is not accessible to MNase enzyme because it is already bounded via the other proteins. So, that's why it is not getting its place and it is not able to cut. But in the case of probe 2, it might be the free accessible portion of MNase because depending on the time, it is cutting and cutting it over. So in a chromosome, which part is the most condensed part? It is the centromere. So the first probe might be representing the centromere of the chromosome. So the option B is true. And next, see, the probe 1 may correspond to the hypomethylated locus of the genome. Depending on the epigenetic modification of the chromosome, if there is any hypomethylation, then the gene would convert into a euchromatin structure. So that will be very much accessible to this kind of MNase enzymes. So that is not happening here. So this is not true. And the last one is saying about the probe 2, which may correspond to an euchromatin locus of the genome. Euchromatin is the most accessible part of the genome, so it might be in euchromatin form because the MNase is getting its free place to sit and cut it. So this D is also right. And the last one is that the option A is saying about the size of the probe 2 is smaller than the probe 1. So here, from this southern blotting result, we can't interfere on the size or the length of the probes. So this is not applicable in that case. So the option B and D means the second option is right. So in this question, the DNA samples were taken in several tubes and each tube was treated with the several chemical agents. Thereafter, they got this kind of result from separate tubes, and you have to determine the reason behind this. And here we all know DNA is made up of double strands, where the each single strand is connected via the hydrogen bond, H2 bond. So here, each single strand is actually a chain of nucleotide, where each nucleotide is made by nitrogen base, which connects with the N glycosidic bond with the pentose sugar, and which again connects with the PO4 group, where you can see a phosphodiester linkage with the next nucleotide. And this bond is known as the N-glycosidic bondage. So here, if you want these nucleotides, then you have to break this phosphodiester bond. So nucleotides will go to the breaks of phosphodiester bond. And the next one is the only nitrogenous base. If you want the nitrogenous base, then you have to break this in glycosidic bond. So this goes to this one. And the nucleoside, and this total thing is known as the nucleoside. When the PO4 is attached with this, then it will transform into the nucleotide. So you have to remove the phosphate group to make it nucleoside. And single stranded DNA you will get when there is a breakage in the hydrogen bond. So this is, so option 2 is correct. So this question is about the erythromycin, which is a well-known antibiotic, and it shows bacteriostatic activity. That means it can restrict the growth of bacteria but can't directly kill them. And here we have to know the mode of action of erythromycin. So first we have to see how the initiation of bacterial translation happens. If this is the mRNA and this is 5 prime, this is 3 prime. So in the case of the translation, there the ribosome will be here. This is the 50th subunit, and this one is the smaller subunit, that is the 30th subunit. And in the case of 50th subunit, there are 3 separate chambers are present there. So the first chamber is known as the A site, this one is the P site, and the last one is the E site. So A site is the direct entry point of the amino acid tRNA, and P site is known as the peptidal site, and the E site is the exit point of the RNA of the prokaryotic system. The first tRNA, the first amino acid tRNA is the F-mate tRNA. Here is an exception. Only this tRNA goes inside via this P chamber. So here the F-mate tRNA first comes to the P site, that is via the decoding process. What is the decoding process? Because this fmate tRNA has the anticodon site and it binds exactly with the codon site, which is the initiation codon that is present here only, and the subsequent codons are present like that, that and that. So here, when the fmate tRNA first comes to this P site and stays there like this, and the next amino acid tRNA comes through this way from this A chamber, and depending on this codon, the anticodon, the amino acid tRNA having the anticodon site, it comes and binds here. And here the next step happens, that is known as the transpeptidation. Here, so here is an amino acid and there is another amino acid, then there will be a linkage between them that is known as the transpeptidation process. And here, to continue this process, this ribosomal complex should go on that way. So this is known as the translocation. It happens in each step. It happens for a single codon. It goes towards that, towards this 3' direction to catch the next codon. But here, if the erythromycin is present, so that will bind to the 23S ribosomal subunit of this larger subunit of this 50S larger subunit. So, and that will make a reversible blockage at this exit site. So here, even there one transpeptidation has happened, but still to go on this process, it is not possible, because here the erythromycin is present and it has make a block at the exit site. So this 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 amino acid tRNAs can't go further on that direction, because this so that these chambers will not be free to welcome the next amino acid tRNAs. So this process will no longer be available. So here the erythromycin inhibits the protein synthesis at the step of elongation. Yeah, it stops the elongation but not the initiation. So this is right. And here it allows the translation of the first few codons. Yeah, we have seen the translation will happen for the first few codons. So that is right. But here it is saying that it prevents for the formation of even the first peptide bond. That is not true. We have seen there is a peptide bond formation, and the rest are not true. That erythromycin inhibits the formation of translation initiation complex. That is not true. The initiation was there, but the elongation was restricted.